name is Precious Kumi, and my presentation is on a comparative assessment of immune responses against two COVID-19 vaccines used as boosters in Ghana. So basically, COVID-19 boosters stimulate the immune system to generate a heightened response against COVID-19. Now, my research focused on looking at the duration of boosted immunity by assessing T cell and antibody responses to two COVID-19 vaccines that we had used as boosters. Now, what we did was we leveraged the vaccination platform here in Ghana, where people had been vaccinated with the Pfizer, Johnson & Johnson, AstraZeneca, Moderna, and then the Sputnik 5. So, as part of our study, we recruited people who had been fully vaccinated and then enrolled them in our study. So what happened was, on the day of sampling them, we took their demographic data, clinical history, vaccination history, and then gave them the booster shots. Now, by the 3rd month, we came back to come and sample them again by the 6th month, and then at 9 months. We extracted PBMCs from the whole blood samples that we took, and then tested them for interferon gamma responses. And then for the ELISAs, we tested their plasma against synthetic SN spike and then nucleocapsid peptides, and also against the full-length proteins. Now, what we realized from our results was that antibody responses, as a result of the booster vaccines that we gave, increased significantly by the 3rd month. But then these responses waned as we go down the timelines. So by month 9, antibody responses waned, but then not that significantly. So, but basically, our booster immunity worked very well within the first 3 months. Now, we're not only interested in the number of antibodies that were produced. We wanted to know which aspects of the spike protein were responsible for the antibody responses that we were seeing. So, we used 2 bioinformatic tools, ALBitope and then BICEPS, to predict regions within the spike protein that overlapped by at least 16 amino acids. Sorry, that overlapped by 16 amino acids. And then tested these peptides for antibody responses. Now, from our results, the receptor binding domain, which is represented here by, so we tested 32 peptides, but then these are just the result of 4 peptides. So, but then the results here showed that within the receptor binding domains, there was variable antibody response, where within some of the regions within the receptor binding domain, antibody responses decreased across as you move down from day 0 through to month 3, month 6, and then month 9. But then there were other regions within which antibody responses increased. Now, there were also other regions within the spike protein, such as within the end terminus, where there were increases in antibody responses. Where there was the worst production of antibodies against the spike protein was at the S1 and then the S2 cleavage site. Now, antibodies within that region decreased significantly from month 3 all the way through to month 9. All right. So we're also interested in knowing whether natural infection played a role or contributed to the amount of anti-spike antibody titers that we were seeing. So we measured responses to nucleocapsid protein, and then during our follow-up. But then what the result here is showing is that natural infection did not contribute to the protective antibodies that we're seeing. And then my figure 4 here is just a representation of T cell responses. So this is at the 3rd month of analysis of vaccine-induced T cell responses. This data here is representative of that. Now, the T cell response also just mimicked whatever was happening with the antibodies, in the sense that T cell responses also waned as you move along from day 0, month 3, month 6, and then month 9. Now, we also were interested because in Ghana here you had received 5 boosters, and then you had received 5 types of vaccines that had been used for primary vaccination. We were interested in knowing, and then we gave 2 boosters using the Pfizer and the Johnson & Johnson. So we realized that we could get we could heterologously boost and then heterologously boost and then homologously boost. So we were interested in knowing between the two vaccination regimens which one worked better. Now the result here is showing that there was no significant difference in the performance of either strategies or either vaccination regimens. But then the means, but then it's just the results that you see here just sort of just suggest that slightly heterologous vaccination strategy may be better than homologous, but then the difference is not significant. Yes. Now we're also interested in the vaccination platforms, whether between an mRNA vaccine and then a viral vector vaccine, which of the platforms works better, right? So what we realized was that there was within 3 months after booster vaccination, mRNA vaccines elicits a very strong immune response or induces higher antibody titers compared to viral vectors. But then if you move down the months, the viral vector vaccines seem to perform better. So our conclusion here was that while an mRNA vaccine might induce significantly robust antibodies within the first 3 months, viral vectored vaccines may confer better long-term protection. So essentially, this is the work that we did evaluating the longevity of boosted immunity with a focus on antibody and then T-cell responses, and also trying to evaluate between homologous and heterologous booster vaccination strategies, which one was the better one. Thank you.